Are we all back? Oh, goody. Okay. Let's do it. Let's talk about the lab. And so this week's lab, as we've been talking about in lecture, is on biological molecules. Does she call it macro? No. No, detection of biological molecules. And it is exactly that. It's how do we detect the biological molecules we've been talking about? Proteins, carbohydrates, lipids, which we will get to in the lecture on Thursday, and nucleic acids, minus that one. We actually don't have a laboratory method that we can do at Mira Costa or any community college really to detect nucleic acids. That you have to kind of do a different, longer type of technique. But when we talk about these chemical indicator reagents that are going to change colors, we don't really have one for nucleic acids. So we're going to be actually detecting the three classes that I mentioned, the three categories there, but not just carbohydrates. We actually can detect simple carbs, simple carbohydrates like monosaccharides and disaccharides that we've looked at. We can detect those, and then we can also detect longer chain polysaccharides like starch and like glycogen, those polysaccharides that we looked at today. So those are going to be the four types of molecules that we're going to look at and detect. And so for this mini lecture, I want to go over the chemical reagent, we call it, or it's a liquid substance chemical that we use in the lab for any experiment. We call it a reagent, but we're going to use different reagents to detect different classes of molecules. And we're going to see, like I said, for this lab, the purpose is to look at a solution such as glucose or honey, egg white, even water. Just to analyze it by doing an experiment on it, adding a chemical reagent to it, and looking for a color change. And a specific color change will indicate if that biological molecule is present in that substance, in that solution, or that thing we're looking at, the egg white, the honey, whatever. So let's talk about how we go about doing this, okay, for each class. So we have four that we can examine, right? We have simple carbohydrates. So this would be I'm just going to abbreviate because I got a smaller whiteboard. Turn this on. Simple carbs. This is our monosaccharides, our disaccharides. Mono and disaccharides. Actually, so let me, okay. Okay, let me do. So on your paper, you might have to, I'm trying to think, I probably can't. You're gonna do a chart kind of thing with 3 columns. I think I can only fit 2 columns on my board here. So I'm just gonna do 2 for now. This column is gonna be the type of biological molecule. I only have 1. Let's do biological molecule. And then this column is going to be the reagent that we use, the reagent used to detect it. The third column, which I'm gonna probably end up erasing this one. The third column is gonna be the color change for the presence and absence, might as well add that, of that molecule. So if, for example, a monosaccharide like glucose or fructose is present in our solution that we're looking at, in our liquid or substance, whatever it is, it will change a certain color specific to the chemical that we use for the test to detect for simple carbohydrates. Okay? I don't know if that makes sense, but I will explain it again in a moment. Okay. Let's go through our biological molecules that we are going to detect. Simple carbs, and then of course, complex carbs, as we call them. These are our longer polysaccharide chains. These are our polysaccharides like starch. So we can detect those. We can detect proteins. Our polypeptide chains, our chains of amino acids that form these proteins, we can detect those in an unknown solution, which is what you do in the lab. Their second part is unknowns, but I'll get to that. Last but not least, lipids. Detect lipids. We haven't gotten into it in lecture yet, but I'll just give you a brief, I'll just tell you that our lipids are our oils, our fats fall under the lipid category. So we can detect those as well. All right, so let's start with our simple carbs. I believe that's the first one in the lab anyway. So the reagent, oh no, it's not first, but that's okay. It's second. Polysaccharides are first. That's all right. So our mono and disaccharides, the reagent used, it's called Benedict's reagent. And I'm going to use blue because it's actually blue. It's a dark blue. It's a liquid. So it's called Benedict's reagent. Okay. So we use Benedict's when we're trying to identify if what we're looking at contains monosaccharides or disaccharides. Glucose, fructose, lactose is disaccharide, maltose, another disaccharide. But we use this Benedict's reagent and we add it to that substance. We add it to that solution. One unique thing, I need a different color to emphasize this. One unique thing to the Benedict's test for simple carbohydrates is that it requires heat. It requires boiling the sample, actually. Heat required. Okay, so if you look at the protocol, I believe I sent you guys the lab. It was that second email. But you'll notice that there's a step for Benedict's and this sugar, the simple carb sugar detection, where you place your test tubes containing your samples in a boiling hot water bath. So that's just an important step required here for this test only. The other tests don't require this heat. Okay. The next reagent that we use to identify and detect polysaccharides is called Lugol's iodine. Let me, I have the colors, might as well, right? I have these new markers. Lugol's iodine is this yellowish, I don't have yellow, but I have this pale orange, close enough. It's a yellow liquid, Lugol's iodine. Okay. So you add Lugol's iodine to each of these, or to the substance that you're trying to detect if any polysaccharides like starch are present. And okay, we'll leave it at that for now. Proteins use a reagent called Biorette's reagent, and it's a blue. It's blue. So Biorette you use to detect proteins. They call it Biorette's reagent. I forget here. Yeah, they do. So Biorette reagent is going to be used for the detection of proteins. So let me see. Sophia, when you say the color, so when I say color change, I'm gonna explain that more clearly in a minute when we look at our third column. But to answer your question, Benedict's is blue, Lugol's is iodine, Biorette is blue. These are the colors of the reagents themselves. And so, as I'll mention this again, when you add it to, let's say you add Biorette to something, to a substance, and it stays blue, it didn't change colors, that means proteins aren't present because Biorette's already blue. Again, I'll go over this again. I just want to first sort of show you that each category of biological molecule has a specific chemical reagent that you use to detect for it. Okay, last are lipids. It's called Sudan red. It's a red dye. The lipid test is a little different. I'll mention it when we get to there. Here they call it's Sudan red, or Sudan, I guess they call it Sudan 3, but it's red. Okay. Sudan 3. It's the red reagent. Okay. Now that we can see what chemical reagent we're going to use to detect each of these molecules, I'm going to talk about the process very briefly here about what we're doing exactly in the lab. If we're looking at a substance and we want to see if there are monosaccharides or disaccharides present in this substance, we're going to add Benedict's reagent to the substance. So let's say we have like honey or egg white or whatever we're looking at in a test tube. We're going to add Benedict's reagent to that test tube. And again, Benedict's reagent is a chemical, liquid chemical. It's in this bottle, it's blue. What we're looking for in this lab is color change. We're looking for a color change. So I mentioned Benedict's is this blue. So what I'm going to change this category to is positive test result. I think that's the best way to do it. So a positive test result, meaning that the substance we're looking for is present. It is there. The honey, the egg white, whatever we're looking at contains simple carbohydrates. If you get this color change. So I'm actually going to do this. Like I said, Benedict's is blue, right? So if you see a color change from blue to actually a variety of colors you can see, but more often than not, a positive test for Benedict's is blue becomes this reddish, orangish, like brick colored orange, reddish, and sometimes green for some reason, basically any other color than blue. Then you have simple carbohydrates in your solution there. So I'm going to write orange, red, green. You see that color change? That sample contains simple carbs. I saw a question here. Yeah, rust, exactly. It's like a rust color, totally. That means, and again, the Benedict's test requires that you boil the sample, or else you won't see a color change at all. Doesn't mean the simple carbs aren't there. You just gotta boil it. So I'll leave that there. That's an important thing with Benedict's test. Okay, same sort of thing with complex carbs. You have a sample, and if you're looking for a complex carbohydrate, you're going to add Lugol's iodine, which is this yellowish, so I'm going to write yellow. It's this yellow liquid. So I'll write yellow. It's like this pale yellow. And if polysaccharides are present, such as starch or glycogen, cellulose, these types of things, the Lugol's iodine turns this very dark blue purple. Exactly. So Gloriana, I'll emphasize that point again in a moment. So thank you. She has a great question. So let's say I add the Lugol's iodine. It's yellow. And then we see this drastic color change to this dark blue purple. So I'm going to write dark blue / dark purple. Very striking change from yellow to dark blue purple. That means we have a positive result, right? The polysaccharides are present. So I'll just tell you right now, we talked about it. Potatoes, bananas, wheat, they have polysaccharides, they have starch in them, right? So if you take Lugol's iodine and just put a few drops on a potato, it's going to turn dark purple, dark blue. It's kind of cool to look at under the microscope. Okay, now to address Glorianne's question. If you don't see that color change, it does not have the molecule we're looking at. Exactly. So if it stays yellow, you add Lugol's iodine, you don't see it change dark blue or purple, and it's still yellow, there are no polysaccharides present. Same with the simple carbs. You do the Benedict's test and it stays that dark blue, then no simple carbs present. So that's a good question. That's what we call a negative result. Proteins, Biuret, it's blue, right? And it turns this very pretty looking light purple, lavender color. So it turns from a dark blue to this light lavender purplish color. Do I have a lighter purple? I don't think so. No. So I'll just write lavender. And lipids are a bit strange because I'm just going to write red. I'm going to write let's write no stain to red. And I'm going to explain that because the lipid test is pretty different. It's very, very different, actually, than the rest of them. And actually, before I explain it, I'm going to go through the lab a little bit, just like how 450. Okay, so just for about like 5 minutes, I'm going to go through the procedure and how we actually do this. And I'll spend a couple more minutes talking about what you do for lipids, because it's a little more complicated and different in a sense than what we do for these three. But essentially with lipids, if you see a red stain, then lipids are present, or these oils, fats, lipids, they are in our substance. Okay. So the first part of the lab, there's two parts. Part A, stain. I'll explain that, what I mean by a stain. It's because we're going to use filter paper, just a piece of paper, and there'll be like a red dye stain on it. This is for the lipid test, but I'll explain that. Okay. All right. So I'll go ahead and just very quickly, for the first three, kind of group them together. It's a very similar process for the first three here. We're using test tubes, and for part A, or part one, known solutions, because part two we're doing unknowns, and identifying molecules and unknowns, which I'll get to. But for part 1, we have water, starch, egg white, glucose, honey, and vegetable oil. So we know what they are. And we're gonna, well, first you're gonna predict what molecules are present in these items, food mostly. Yeah. And then after we do the experiment, you're gonna, record what they actually contain, whether it's simple sugars, proteins, lipids, etc. Okay, so for each of the substances, you're gonna do, or you're gonna watch the video, the data for what happens with each of these tests for each substance. And again, for the first 3, if you're testing for proteins, complex carbs, simple carbs, you're going to be adding these solutions, right, the water, starch, etc., to test tubes. And then to each test tube, let's say we're doing the simple carb test, you would add what reagent? I erased it, but what reagent if I'm looking for simple carbs in all of my known solutions? Benedict's, exactly. You'd add it to all the test tubes, and then for Benedict's uniquely, you would actually, before you see a color change, you gotta place it in boiling water, submerge the test tubes, not entirely, you just put the tips of them, or the bottom part. And you would look for that color change for each test tube. You'd label it, you know which one is in it, which one, and then you could see which test tubes change color. So you would know which substances had simple sugars in them, which ones changed to that rust red, orange, sometimes green color. And then which ones didn't, which ones stayed blue, that dark blue. You do that as well, same sort of thing with test tubes, but different reagents, of course. You'd use the Lugol's iodine for starch and polysaccharide detection, and then the Biuret for proteins. Do the same sort of thing, except of course, no heat's required. You just add it and you can see the color change right away. And it's kind of cool. Okay. Lipids. The lipid test, when you use these substances for the lipid test, is a little weird. But essentially, you're gonna you have a piece of filter paper. It's circular. I'll just draw a very simple sort of diagram. There's 6 solutions, right? And you divide the filter paper, it's a circle, into 6 sections evenly, not like that. Why did that not look even? Let me try that again. Good enough, right? I'm not a math guy. All right. But what you do is you take this paper and with each of the 6 solutions, you put a drop. So you'd have your solutions in 6 test tubes, and you add a drop to each section. So you do a drop there, you have another one for that one, etc. And you would do that, and then you take it to a hot plate and you let it dry. Lipids, remember, it's detecting fats and oils, so they actually don't dry. That's the key here. They stick to the paper. So you take it to the hot plate to evaporate it, because at the end of the lipid test, you actually take the filter paper and you drop it in a dye. That's the Sudan red, the Sudan 3. You drop it in this dye, and if there's anything present on the paper, the dye is going to stain it. And so lipids, being these fats and oils, are the only thing that stay on the paper. So what you do is you'll be adding the solution, drops dropwise to the paper, take it to the back, use a hot plate to evaporate it, speed the process, and you do that 3 times. Yeah, you do it 3 times, and let's see. Okay. So 3 times for, you do all 6, hot plate, then do it again, all 6, do it again, and then you drop it in the dye. And after that, you look to see which one is red. The oil will not have evaporated, and that's the one that has the lipids. And you'll see that stain on the paper for the section where you dropped the lipid or whatever you're testing that has lipids in it. Okay, so how are we doing on time? I really want to end this. So we're good soon. I'm going to wrap this up. Essentially, part B, the only thing, is the same exact thing, truly, except now your solutions are not known. They're unknown. And so you do these tests to all your unknown solutions and identify what's present in these unknown solutions. That's it. So maybe the first one has lipids and it tested positive for lipids and also proteins, but not for monosaccharides or whatever. Okay, yeah, it is fun. In person, it's a lot of mixing and color change. It looks cool, I know. Yeah, so I wish we could do it in person. But one more thing I'll say, the purpose of the first part of the lab is not just to practice the tests, but it's to learn and see what a positive result looks like in person, just to see it, and to see what as well as what a negative result looks like. And we call these a positive control and a negative control. And the purpose of that is for reference, really. And it's a comparison. It's point of comparison. So when you do part 2 and you look at unknown solutions, if you see a color change, compare it to your positive control of that color change, the one that you know is positive, and see if they're the same. Compare them. And then that'll tell you that, yes, it is positive. We also do controls like that to make sure that the reagents are working, that the experiment, we're not doing anything incorrect that might not give us the correct result. But I believe that's all I wanted to say. Yeah. So again, positive control, you will know what the positive result looks like, so that when you do an unknown, you have something to compare it to. You're not just seeing this color change and not knowing by just me writing it what it means, but you can actually compare it and say, oh, that's what positive looks like. So that has the protein, or that has the, and negative, of course, then you can compare and say, oh, that's what it looks like. Questions? Ended at 5 on the dot. Any questions? In table 1, what do the abbreviations stand for? It's the classes of molecules, but I think they're it's protein, sugar, or starch. The person who wrote the lab it should say, well, proteins is okay, but starch, really it's and it's up there on the top too. I'll just say that. So, but starch, aka polysaccharides, and F, fat, but really that's lipids. Fat is not the scientific. So any other questions? All right, then I sent you the labs, so I'll see you Thursday. Cool. Have a great rest of your evening. Thank you guys.